Hi everyone, we're about 5 minutes away from starting today's review session. Just curious right now if everyone can hear me. All right, excellent. So I'm just going to wait a few more minutes. We'll get started right at 1 o'clock. There's about a 30-second lag time between when I say something and when you hear it. So I'm just going to keep an eye on the chat for the next few minutes. And if you have any questions right off the top before we get started, go ahead and type them in. We have 3 other experienced AP biology teachers moderating today, so you're going to have lots of help. All right, is there still an echo? I just muted my computer. I don't know if that helped. Still an echo. Okay, let's see if we can solve this quickly. Hey, so it's saying, I'm getting a message that says that the bit rate, the audio bit rate is not optimal. How did you change the video setting yesterday? Sorry, you're gonna. Okay.
So it's in the settings.
Right click on here.
Or just click on
That? It said 128. But I don't see any options to adjust it. Right? Yeah, okay. Okay. It may just be because I'm here in the room and I'm not speaking directly in the microphone. Okay. All right, cool. Maybe close the door. Okay. Yeah. What they're saying is that it doesn't sound too bad, just like I'm in a big room. So that's probably what it is. Okay. Okay. All right, I'm gonna get started. Yeah. All right, cool. Thank you. Thanks. All right. So it sounds like the echo isn't too bad. I think it could be because I'm not speaking directly into a microphone. I'm speaking into my laptop microphone. So I apologize if it's not perfect sound. I am I'm not using two microphones. I'm only using one. So I apologize if it sounds a little strange. This is my first live stream, so please be gentle. But I'm going to go ahead and get started. Today's review session is on the chemistry of life. My name is Jamie Castle. I teach at Pennsylvania Leadership Charter School in Westchester, Pennsylvania. I teach AP Bio. I also teach AP Chemistry and Human Anatomy and Physiology. If you have any questions about what you hear this hour, after the fact, you can contact me on Twitter at Jamie N. Castle right there on the screen. So. All right, a couple of housekeeping items before we get moving. There are some links below the video, actually several links below the video, and I just wanted to highlight a few things. One link is the slide deck from this presentation. If you want to see a copy of that, save it to your Google Drive. Another is the schedule of the remaining review sessions. So if you want to attend any of the other sessions, you can just click right on those links and they'll take you right to the live stream like today. If you want to go back and watch a stream that you missed, click on those links and it'll take you to the recording. And then also there's a link for the question submission form for the future review session. So optimally, if you submit a week ahead of time, that'll give the review session presenter time to factor your question into the session. So if you can get those questions out, the earlier the better. And the link is there. And then there's a few other ancillary links there as well. All right, so how to use this video most optimally. One, this will be a really nice refresher of the content from early in the course, some of which you probably haven't thought about explicitly in a handful of months. You can also use this video to consider how the material from Unit 1 might be incorporated into this year's AP exam. And then finally, this video will support, but definitely not replace, your own in-depth review of Unit 1 material. This unit usually takes most students, most classrooms, maybe like a week and a half, 2 weeks, maybe a little more to get through. And I'm going to do this in an hour. So I'm going to hit the big points. And again, you can ask lots of questions in the chat. Okay, a few things to keep in mind. I'm going to make a mistake at some point already. In fact, I already did. We have an echo, but I may make another mistake. It's unavoidable. I'm human. So if there's anything that I say that seems unclear, just ask in the chat. Another thing that I wanted to mention is again about the lag. I did mention it at first when I first started the live stream, but there's a lag about 30 seconds between what I'm saying and what you are seeing. So just know that I'm not seeing your comments in real time, but if I see something come up in the chat, I do have the window open over here, and I can, pause and answer it. I certainly will, but you have 3 moderators in the chat with you who can also help you out. The AP questions on the exam, regardless of the format of the exam, is always a combination of content and skills. So another link that I placed down below the video is the AP Biology Science Practices, and I urge you to take a look at those and to think about how those skills can be combined with the content that I'm going to go through today and that all the other teachers are going to go through in subsequent review sessions. And then finally, mental health, especially right now, is more important than exam prep. We're all under a lot of stress. This is a completely unprecedented time for us in history, and we're all figuring out our way day by day, right? So the AP exam is certainly important to you. I know that. That's why you're here. But just keep a check on your mental health. If you're feeling a lot of stress, maybe one day taking some time off from exam prep is a good idea. So just always make sure that you're feeling good throughout this process. Okay, just wanted to say thank you specifically to your moderators who are Hannah Hathaway, Sedate Kohler, and Clifford Nefrata. Please thank them for volunteering their time either now or before you exit the chat later on. Okay, so we're going to get into content now. We're going to start with the first of 6 sections in this unit, and the first section is on the structure of water and hydrogen bonding. And the objective for the structure of water and hydrogen bonding in that course and exam description document that I mentioned before, and that's linked down below the video, this objective is explain how the properties of water that result from its polarity and hydrogen bonding affect its biological function. So it's not just enough to know what the properties of water are, you have to know why they happen and also how it helps water to be such an important part of sustaining life. So let's get into that. So here's the structure of a single water molecule. Water is a polar covalent molecule. It's composed of one hydrogen atom and two hydrogen atoms and one oxygen atom. There's my mistake already. And because oxygen is a very electronegative atom and hydrogen is not, there's an uneven distribution of charge in this molecule. And we can see these little symbols here that look kind of like D's. So we have this D with a minus up here. That means there's a partial negative charge near the oxygen and partial positive charges, these D pluses, down here near the hydrogen. Oxygen also has two additional lone pairs of electrons on it. So you're going to have more negative charge near the oxygen atom and more positive charge near these hydrogen atoms. The two lines that we see here represent our covalent bonds, and we know that that means that there are electrons being shared. So oxygen is sharing one electron with this hydrogen, and this hydrogen is sharing its one valence electron with the oxygen. So this structure of a single molecule, let me take a drink of water 1 second. This structure of a single water molecule allows many water molecules to engage in something called hydrogen bonding. Now, this is a little bit of a misnomer. It's not really a bond. It's an intermolecular attractive force, or an IMAF. And you may remember those from your chemistry class. So there's a number of different kinds of IMAFs. You might remember dipole-dipole interactions, or ion-dipole interactions, or induced dipole interactions. Well, hydrogen bonding is one of those types of interactions, and in fact, it's the strongest type. And this gives water some special properties. So a hydrogen bond only forms under specific circumstances, and that is the attraction is between an electronegative atom, nitrogen, oxygen, or fluorine, okay, and a hydrogen that is also bonded to a nitrogen, oxygen, or fluorine on another molecule. So when we look over here at these water molecules that are hydrogen bonded to one another, we see an oxygen up here in the top water molecule attracted to a hydrogen in another water molecule that is also bonded covalently to an oxygen atom. So this is what is essential for a hydrogen bond. I threw another molecule down here that has a whole bunch of hydrogens in it, but these hydrogens are not going to form hydrogen bonds because they're not bonded to another electronegative atom. Carbon's not electronegative, okay? So it's not that any molecule that has a hydrogen on it can be involved in hydrogen bonding. There are special circumstances. So when we look at this diagram of hydrogen bonding, we see that each water molecule can form hydrogen bonds with 4 other water molecules. One for each of the hydrogen atoms that's covalently bonded in the molecule, and also one hydrogen bond for each of those lone pairs of electrons that are on the oxygen atom. All right, so that's how hydrogen bonding works. And hydrogen bonding gives water on a larger scale a number of special properties, 2 of which are cohesion and adhesion. Because water molecules are polar, they will hydrogen bond with one another, okay? And that's called cohesion. And that's the reason for high surface tension. So if any of you have done the water on a penny lab earlier this year, that's the property that you were testing. When water is pure, those water molecules are hydrogen bonded to one another, highly attracted, and so there's high surface tension. You can get a lot more drops of water on the top of a penny than if you put some soap in there. Once those soap molecules get in between the water molecules, they interrupt the hydrogen bonding, and then there's far fewer water droplets that you can get on the top of that penny. Another property of water that's supported by hydrogen bonding is adhesion. So this is when water molecules are attracted to other polar molecules besides water via hydrogen bonding. So we look at the image over here to the right, we see an example of this, transpiration of water in plants, specifically in xylem. There is cohesion happening, Okay, we can see that the water molecules are kind of following one another up the path of the xylem, but we also see right here in this little blown out image that a water molecule is attracted to the polar components of the cellulose in the walls of the xylem. Okay, this isn't the only example of adhesion, it's just one of them. All right, so let's look at some more properties of water. High specific heat of water is another property. That means it takes more energy to increase water temperature. Okay, it also takes, it requires more removal of energy to lower the temperature of water. Um, I'm sorry, I was just reading the chat. So what that means is that water ends up being a little bit of an insulator of temperature. We won't have crazy wild swings in temperature in water the way we do perhaps in the desert, right? There's huge increases in temperature during the day and then wild drops in temperature at night because the desert is dry. When you live closer to the coast, the swings in temperature are less extreme. That's because of this high specific heat of water. There's also evaporative cooling as a property of water. This means that the highest energy molecules will evaporate first, and that cools the surface of the remaining liquid. This is why we sweat to cool down. Ice also floats, and this is an important property, right? Because if ice sunk, then it would kill everything in a body of water in the wintertime. So solid ice is less dense than liquid water, and we see why over here. As water moves from the liquid phase where the hydrogen bonds are a little bit weaker and the water molecules can move more fluidly around one another into the solid phase where those hydrogen bonds really lock in place, it actually pushes those water molecules a little farther apart from one another, lowering the density and allowing ice to float above the surface of the remaining liquid water. And then finally, water is a versatile solvent. Because those molecules are polar, they can dissolve other polar covalent solutes and also ionic solutes like salts. And we see this, for example, in our cells, which are an aqueous environment. Okay, so that was the first section. Now we're going to move into the second section on the elements of life. This section's objective is to be able to describe the composition of macromolecules required by living organisms. All right, this section's kind of short. So most organic molecules are composed of rings or chains of carbon bonded to hydrogen, oxygen, and or nitrogen. These are covalent compounds. Water is not an organic molecule because it doesn't have carbon in it. It's essential to life, but it doesn't fit that definition of organic. So biomolecules or biological macromolecules are a subset of organic molecules synthesized by living organisms. And we see that list right here: carbohydrates, lipids, proteins, nucleic acids. So the compositional elements of carbohydrates are carbon. Oh, sorry, I'm just seeing a question. What do you mean water can dissolve things other than salts? Did I say that? Okay, I'll keep an eye on the chat, but I'm going to continue. So carbohydrates are made up of carbon, hydrogen, and oxygen. Lipids are also made up of carbon, hydrogen, and oxygen, but in phospholipids, which we'll talk about, there's also some phosphorus. Proteins are made primarily of carbon, hydrogen, oxygen, and nitrogen. There's also some sulfur found in some of those variable R groups, again, which we'll talk about. And then finally, nucleic acids are made up of carbon, hydrogen, oxygen, nitrogen, and phosphorus. All right, moving into the third section. So we'll now spend a little more time in these subsequent sections, especially 3, 4, and 5. So in section 3, the introduction to biological molecules, your objective is to describe the properties of the monomers and the type of bonds that connect the monomers in biological macromolecules. All right, so I'll just come back to the question about water dissolving things other than salts. Water can also dissolve polar covalent solutes, which are not salts. Yes, exactly. Like Hannah just said, like glucose. Glucose isn't a salt, it's a covalent compound, but it does dissolve in water. Okay, going back into this section on introduction of biological molecules. So let's differentiate between monomers and polymers. Polymers are large molecules made up of many similar molecular building blocks. Monomers are the building blocks. So in a very simple summary, we see that carbohydrates are the polymer that are made up of saccharides. Proteins are the polymer that are made up of amino acid monomers. Lipids are the polymer that are made up of, and this is an oversimplification, some lipids are made up of fatty acid monomers, but lipids are kind of a special case. Some lipids don't have monomers really. Some have monomers plus, some have fatty acids plus something else. So I oversimplified a little bit here, but if you really want to put this into a simplified chart, this is a good way to think of it. And then nucleic acids are the polymer made up of nucleotide monomers. We can also see in the image over here, we have a bunch of single small building blocks. And through the process of polymerization and dehydration synthesis, which we'll talk about in just a second, we are able to obtain a much larger polymer. Okay, so let's talk about how monomers are joined together and also how they're broken apart. So I just mentioned dehydration synthesis. These are also known as condensation reactions. They join two monomers together into a polymer, and in the process, a water molecule is removed. So when we look at the diagram here, we're starting on the bottom with our monomers, and as we move toward the up arrow, there's a water molecule removed, and then we see that for each water molecule removed, there is a covalent bond formed. And we'll talk about the specific names of those bonds and what's involved in each of those different dehydration synthesis reactions when we talk about each of the macromolecules specifically. If we're going to go in the opposite direction and we want to break a polymer apart, we have to add water. So if we start up here with our polymer and we move down toward these broken up monomers, we have to put a water molecule in for each individual covalent bond that we want to break. Okay, so same objective for section 4, describe the properties of the monomers and the type of bonds that connect the monomers in biological macromolecules. But now we're going to dig into those 4 categories more specifically. We're going to start with carbohydrates. So the main functions of carbohydrates are to provide energy, for example, in glucose, in glycogen, which is a polymer of glucose molecules, and in starch, which is another polymer of glucose molecules. The difference between glycogen and starch is that glycogen is used for energy storage in animals, and starch is used for energy storage in plants. Carbohydrates can also provide structural support, for example, in cellulose and in chitin. The monomer of carbohydrates, like I already mentioned, is the monosaccharide. These are chains or rings, and most often we see them in chains or rings of 5 to 6 carbons. You can have them in as few as 3 or as many as 8 carbons, but most of what we're seeing in AP biology are these 5 or 6 carbon sugars. You know a monosaccharide when you're reading its name because it ends in ose, so glucose, fructose, sucrose, etc. Right up here, we see what the ring and chain versions of the same monosaccharide look like. So over here, this is our chain version. And I tried to find a better image here that actually had the C's at all these cross points, but I couldn't find one. So just know that at each of these cross points, there is a carbon atom. I don't know that every one of your classes has gone into what this like simplified modeling actually shows. If we look over here on this side, now this is the ring version of that same monosaccharide. So again, over here at all these points, these are also carbons. But if you compare the left to the right, it's the same number of carbons, oxygens, hydrogens. It just looks slightly different. Okay, polysaccharides are our polymers, or otherwise known as complex carbohydrates. The bonds between monosaccharides are called glycosidic bonds, and we'll come back to that in a little bit. I'll show you exactly how those glycosidic bonds are formed. The presence of oxygen makes some carbohydrates polar enough to dissolve in water, but not necessarily all of them. Okay, so it's not a universal. It's not a universal across the board, always polar or non-polar situation with carbohydrates. I do have a diagram down here of 2 carbohydrate monomers, 2 monosaccharides, going through a dehydration synthesis reaction to form a glycosidic bond. Here we see the water molecule that's lost, and we'll look again at this a little bit later on. Over here, I have an example of just a piece of cellulose. You can see that it's very complex. There's branching between the linear chains, so that's one of the complex carbohydrates. Okay. All right. So moving into lipids. So the functions of lipids are energy storage, structure, and they also make up a portion of the ligands in the form of hormones. So when we talk about steroids and estrogen, progesterone, um, testosterone, for example, these are lipid-based molecules that serve as chemical signals in signal transduction cascades. So all lipids are nonpolar and therefore they do not dissolve in water. There's one exception, which is the hydrophilic head on a phospholipid because there's a phosphate group in there. And we'll talk about that, but otherwise all lipids are nonpolar. There are three types of lipids. Triglycerides, or fats, they're formed from 3 fatty acids and a glycerol molecule. Phospholipids are formed from 2 fatty acids plus a glycerol and a phosphate group. And steroids are formed from 4 fused carbon rings. So up here we see our triglyceride. This is our glycerol molecule here. It is bonded to 3 fatty acids, which are these long hydrocarbon chains. This here, both of these, are a phospholipid. This is just a different version of what this looks like over here. This is a little bit more detail. This over here is referred to as a space filling model. So we can see the glycerol molecule here, again, the 2 fatty acids, and here's our phosphate group. And then over here is our example of a steroid, and here are our 4 fused carbon rings. Okay. All right, moving on to triglycerides. So I want to differentiate between a linear carbohydrate, a glycerol, and fatty acid because these 3 things are all chains of carbons. So when we look at, for example, this glucose molecule, we see that it's 6 carbons in length. I mentioned before that we also in AP biology see carbohydrates that are 5 carbons in length. In fact, when we talk about nucleotides, we'll see those 5 carbon sugars that are part of those monomers. And we also have a double bond to an oxygen up here. This aldehyde group up here is a signature characteristic. That's the phrase I was looking for. Signature characteristic of carbohydrate. A glycerol molecule is a 3 carbon sugar. Each one of these carbons has a hydroxyl group on it. Okay, so a little bit different here. Half the length and also no aldehyde group. A fatty acid has a carboxylic acid group on the end. This over here, the C double bonded to the oxygen, single bonded to the hydroxyl group, but the entire rest of the fatty acid chain is going to be carbons and hydrogens. So those are just some features that differentiate those from one another. So fatty acid, like I mentioned, is a long hydrocarbon chain with a carboxyl group on the end. The bond between glycerol and a fatty acid is a covalent ester bond. So we see that here, that the hydrogen from the glycerol and the hydroxyl group from the fatty acid are what forms the water molecule that gets removed in dehydration synthesis. And then what forms is this ester. So this is actually flipped from the orientation here. But this is what you have, a carbon double bonded to an oxygen. That carbon is also bonded to the hydrocarbon chain or the fatty acid that's represented by the R, that's this up here. And then this oxygen and this R prime group represent the glycerol. Okay, let's see, where did I leave off? Okay, so I want to differentiate now between saturated fatty acids and unsaturated fatty acids. Saturated fatty acids have all single carbon-carbon bonds. They are solid at room temperature. So think of, for example, like the fat on a steak. That's a saturated fat. It's solid at room temperature. And the reason why it's solid at room temperature is because there are all single carbon-carbon bonds. You can pack a lot of saturated fatty acids together. And so as they pack together more closely, they form a more solid structure. Unsaturated fatty acids have 1 or more double or triple carbon-carbon bonds, and they're liquid at room temperature. So we see here, this is an unsaturated fatty acid. It has a kink in it. That kink is formed by that double or triple carbon-carbon bond. You can't pack as many unsaturated fatty acids together in a given amount of space. And so these are less dense and they are therefore liquid at room temperature. So think of any of your oils, for example. These are unsaturated fatty acids. When you think about descriptions of foods and they're talking about monounsaturated versus polyunsaturated, that's just how many unsaturated fatty acids there are in those triglyceride molecules. I'm going into a little bit of extra detail here. That's probably a little bit outside the scope, but just trying to tie some of this information to things that you might more immediately relate to. Okay, moving on from triglycerides into phospholipids. So phospholipids are amphipathic molecules. That means that they have both, they have both a hydrophilic region or a water-loving region and a hydrophobic or water-avoiding or water-hating region. The phosphate group that we see here has a negative charge on it. That's what makes this region hydrophilic. This is a polar region. It is willing to interact with other polar molecules, including water. The fatty acids, just like we talked about on the last slide, are nonpolar. They're hydrophobic. So phospholipids are a major component of plasma membranes, and Lee Ferguson will go into this in more detail tomorrow when she does her session on the structure and function of cells. But I did want to throw this small reference in here just to show you and remind you how the phospholipid bilayer orients itself. Those hydrophobic fatty acids point toward one another to avoid that polar environment of the extracellular space and also of the cytosol within the cell. The more hydrophilic polar regions of the phospholipids orient themselves out toward those aqueous environments, both inside and outside the cell. All right, moving into proteins. So up here we have the amino acid. This is our monomer or our building block of proteins. Proteins have so many functions. Three of the main functions are structure, signaling. They can also be ligands in signal transduction cascades. They're also part of the signal transduction cascades. And they're also, proteins are also play an important role in the catalysis of different biochemical reactions. Most enzymes are proteins, so they're made up of these amino acids. So amino acids have directionality. That means that they're not, you can't flip them around and they do the exact same thing or that they look the exact same way. There is an amino terminus on one side, which is a nitrogen covalently bonded to two hydrogen atoms. And then there's a carboxyl terminus on the other side. This is our carboxylic acid group, like I mentioned before. A carbon double bonded to an oxygen, single bonded to another oxygen, and that oxygen is single bonded to a hydrogen. We also have a hydrogen bonded to the central carbon on the amino acid, and then we have a variable R group. You don't have to know the names of the different R groups, but you should know that these R groups have different chemical properties. We can see here, and this image is a little bit small, so I encourage you to maybe open up the slide deck and click on the link here to see a bigger version of this image, but this image will show you the 21 different amino acids that are common in living things. You'll see that some of these have side chains, electrically charged side chains. Some are positive, some are negative. Some have uncharged side chains. Some are specifically hydrophobic, and some have some special features like a sulfur atom, for example. So based on these different chemical properties of the R groups, this helps determine the chemical properties of the amino acid as a whole. And so the interactions of these R groups are going to play a big role in those levels of protein structure that we'll talk about in just a few minutes. Okay. All right. Before we get into more detail with each of these macromolecules, I just want to hit the last one here, nucleic acids. So nucleic acids encode biological information in sequences of nucleotide monomers. So these are DNA and RNA. Each one of these nucleotides has three specific structural components: a 5-carbon sugar, which we see here in yellow, and that's either going to be deoxyribose in DNA or ribose in RNA. There's also a phosphate group or multiple phosphate groups, which we see right here, and then a nitrogenous base, which is indicated here just by the word base in blue, but then over here we see the 5 different possible bases that we learn about in AP biology. So the purines here, adenine and guanine, they have double ring structure, and the pyrimidines, cytosine, uracil, and thymine, these have single ring structures. Those will covalently bond right here. DNA and RNA differ in structure and function, and we'll talk more about that in just a little bit. But I did throw this image in over here just to highlight the really most obvious difference between DNA and RNA, which is that DNA is double-stranded and RNA is single-stranded. All right, we're going to move into the structure and function of biological molecules. So now the objective changes a little bit. Now the objective is to explain how a change in the subunits of a polymer may lead to changes in the structure or the function of the macromolecule. So as I'm going through this section and I'm talking about these different subunits, think about how if the subunits change, how the structure of these molecules might change. Okay, complex carbohydrate structure. So polysaccharides, or complex carbohydrates, are composed of linear chains of sugar monomers, or monosaccharides, and they're connected by these covalent glycosidic bonds. So here we have two monosaccharides, glucose and fructose. Which nitrogenous bases have two rings? I'm going to let the moderators answer that question. Glucose and fructose are two different monosaccharides. We find them in different sources. They are both sugars. They both taste sweet, but we find fructose more in fruits. We find glucose more in, for example, like I said, glycogen and starches and cellulose. When glucose and fructose undergo a dehydration, synthesis, or condensation reaction, they form a glycosidic bond right here. We see this oxygen that tips us off that this is a glycosidic bond between two monosaccharides. Where did that glycosidic bond form? Where was the water taken from? Well, we have the hydroxyl group here on the glucose that is taken out, and also the hydrogen from a hydroxyl group on the fructose. These two join together to form a water molecule. They're removed, and what's left is this one oxygen here on the fructose, which then joins together with the carbon here on the glucose. This forms sucrose. Sucrose, also sweet, but has slightly different properties because now there are two monosaccharides joined together. And again, sucrose, slightly different source. Sucrose is table sugar. Carbohydrate polymers, real big polymers, can be linear or branched. So I threw in two examples of that over here. Amylose is a linear polysaccharide. This is a component of starch. Amylopectin is a branched polysaccharide, also a component of starch. All right, moving on to protein structure. So this, in my opinion, is some of the more challenging material in this unit, just keeping straight what the different levels of protein structure are. So proteins, at their most basic level, comprise linear chains of amino acids connected by peptide bonds. So we see a peptide bond being formed up here. These are formed by the hydroxyl group from the carboxylic end, or the carboxyl end of one amino acid, and one of the hydrogens from the amino end of the adjacent amino acid. Those two groups form a water molecule, which we see over here. It goes away, and then the carbon and the nitrogen of the adjacent amino acids will join together in a peptide bond, a covalent bond. Put this image in down here because I wanted you to see how, when we join many amino acids together, it just forms a long linear chain. So this is the backbone of this polypeptide. This is our nitrogen, our amino end of this polypeptide, and then we have two carbons. This is serine, like we see down on the bottom. Then the next amino acid is histidine. Here's our nitrogen and two carbons. Then cysteine, here's the nitrogen and two carbons, so on and so forth until the end of the polypeptide chain where we see the carboxylic end of the chain. All right, so to recap the levels of protein structure before going into them in a little bit more detail. Primary structure is quite simply the sequence of the order of the amino acids. So we see in the image here, this is acetine, glycine, phenylalanine, so on and so forth. This order will dictate the kind of folding that we then see in the secondary, tertiary, and quaternary structures of protein structure. In the secondary structure, we see the folding of this primary amino acid chain into alpha helices, which we see here, looks like a phone cord, or beta pleated sheets. And this folding happens due to hydrogen bonding between the polypeptide backbone atoms. So we'll take a look at that in a little more detail on the next slide. Tertiary structure forms the overall three-dimensional shape of the protein. So all of these alpha helices and beta pleated sheets now form together into more of a globular shape. And this shape is formed by non-covalent interactions between the atoms in the R groups. Again, we'll go into this in more detail in either the next slide or the one after that. But this folding often minimizes free energy. This is why the proteins fold the way they do. The lower energy overall in the molecule, the more stable the more stable that molecule is going to be. And then finally, quaternary structure is just the joining of several tertiary structure proteins together. So this example of quaternary structure here is hemoglobin. It's made up of 4 different polypeptide units that we can see in the different colors. Each of those polypeptide units has a tertiary structure of its own. This protein only has a quaternary structure because it's made up of multiple subunits. If a particular protein doesn't have multiple subunits, it won't have a quaternary structure. All right, so let's dig into secondary structure, as I mentioned. So we're looking at alpha helices and beta pleated sheets kind of in gross form here, and then more in fine form in these 2 diagrams here. So when we zoom into our alpha helix, we see these dotted lines between the the hydrogen bonded to the nitrogen of one amino acid and the oxygen on the carboxylic end of another amino acid. This is a hydrogen bond. We see another hydrogen bond here and here, okay? That's the interaction that forms an alpha helix. Those same interactions form beta pleated sheets. So we see those hydrogen bonds here between oxygens and hydrogens bonded to nitrogens across the span of this beta pleated sheet here. Okay, moving along to tertiary structure. Tertiary structure, again, is the overall 3D shape of the protein. So when you're looking at this image and you see this long, windy, red chain, know that this is made up of alpha helices and beta pleated sheets. This is not just simply the primary structure of the protein. Okay, that's one thing that this image doesn't do a super great job of showing. But this protein has already folded into its secondary structure, and now, as a result of that, the R groups, the different atoms in the R groups will start to interact with one another. So when I talk about non-covalent interactions, I'm talking about things like hydrogen bonds. We'll still see those. We'll also see disulfide linkages between cysteine groups, for example, that have sulfur in them. We'll see hydrophobic interactions between R groups that are hydrophobic, much like we saw the way that the phospholipids orient themselves where the hydrophobic tails all face the inside of the phospholipid bilayer. That's the same idea here. We also see ionic bonding, and some of you who remember chemistry may go, wait a minute, I thought ionic bonds were like the strongest bond, and why are we calling them non-covalent interactions, and why are we suggesting that maybe they're weak? Ionic bonding is not strong in water, and remember, proteins are being synthesized and folded in the aqueous environment of the cell. So these ionic bonds, they're not really bonds, they're just attractions between ions of opposite charge. Okay, they're not forming those really strong ionic bonds that you think of when you think of things like table salt. So this is tertiary structure. Again, non-covalent interactions specifically between atoms in the R groups. And then finally, this quaternary structure. Again, we see 2 different subunits with tertiary structure being joined together, again, by these non-covalent interactions. Okay, so ionic bonding, hydrophobic interactions, disulfide linkages, hydrogen bonding, these can all contribute to quaternary structure as well. Okay. So let's talk a little bit about what happens to these proteins with these multiple levels of structure in different environments. Mainly the secondary, tertiary, and quaternary structure of proteins can be broken down or denatured by changes in temperature and pH because of the weaker nature of these non-covalent interactions. It's really going to take a lot to break apart the covalent bonds that make up the primary structure. It does not take as much to break apart these non-covalent interactions. So if temperature gets too high, for example, and this molecule starts to vibrate more, because that's what happens when the temperature increases, those non-covalent interactions will start to break, much like when we see a substance move from the solid to the liquid to the gas phase, right? It's because those interactions between the molecules are getting weaker. When this happens, there's a structural change that occurs. We see here over on the left in this image, this is a normal structure, quaternary structure protein. When it's exposed to heat, notice now that that has loosened. We've lost some levels of protein structure. We've lost definitely the quaternary structure, and it looks that we've lost the tertiary structure as well. So, and this is a little bit erroneous because these subunits won't necessarily be joined end to end like this, but it still gets the point across that these changes in temperature or these fluctuations when the pH becomes too high or too low can also cause this denaturing to happen. When you have this structural change, you're going to have a functional change. And the reason why is because proteins fulfill their purposes because of how they're shaped. For example, enzymes in their active site, there are certain atoms, certain functional groups inside that active site that allow that enzyme to interact with the substrates to catalyze a particular reaction. If the enzyme is no longer holding its shape, now it no longer has that pocket or that active site to allow that reaction to move any faster. And so function and structure are so closely related here, and it can be very clearly illustrated with proteins. Okay. Nucleic acid structure is made up of linear sequences of nucleotides. The ends of these nucleic acids are defined by 3' and 5'. We see 3' hydroxyl ends and 5' phosphate ends on our molecules. So up here is our 5' end. It's not showing the phosphate. The phosphate would be up here. Actually, this says 5 to 3, but this is our phosphate end. So this diagram is incorrect. You know what, guys? I apologize for that. I did not notice that when I put that in here. I liked this image because it showed the structure of these molecules, but the 5 and the 3 are in the wrong place. So 3' hydroxyl end down here, 5' phosphate end down here. Okay. And oh no, I'm confusing myself. Okay, here's another mistake, and I apologize for that. We're talking about the phosphate, the ends of the sugars. So, no, I had that right. I did have that right. Okay, so here's our phosphate end, and our hydroxyl end is down here. Yes, okay, my apologies. So during DNA and RNA synthesis, and when you watch the review session for Unit 6 on molecular biology, they'll go into more detail on this, but during DNA and RNA synthesis, nucleotides are added to the 3' end of the growing strand. Okay, so I threw this image in here of transcription happening, and you can see that the direction of building of this RNA strand is originating with 5 and then building, extending the 3' end. So those new nucleotides will be added to this 3' end. Okay. Yes, I'm seeing now, isn't it showing how it's synthesized? Yes, I believe that that's perhaps what's being shown. Okay. All right. So a little bit more on nucleic acid structure. DNA is structured as an anti-parallel double helix. So when we look at this diagram here, Okay, this is much more clear. Here's our 5' end here, and here's our hydroxyl 3' end here. But if we look at the complementary strand, our phosphate 5' end is here, and our hydroxyl 3' end is here. Okay, each one of these strands runs in the opposite direction. And then in the middle, our nitrogenous bases pair together via hydrogen bonding. So adenine and thymine pair together via 2 hydrogen bonds, which we see right here. And then cytosine, and guanine pair together with 3 hydrogen bonds. So we see that here. We also see guanine-cytosine pairs here, and we see another adenine-thymine pair here. Okay, this is our backbone here, and again here. Okay, so yeah, for 5' and 3' end, look at this image. This image is much better than the one that I put on the previous slide. My apologies again for that. All right, so that was the deep dive into the 4 major biomolecule groups. Now there's just a little bit more that this unit covers on nucleic acids. So in our 6th and final topic, the objective is to describe the structural similarities and differences between DNA and RNA. So, here's this image again, my favorite. I will change this and put that other image in here, the one that I like better after the live stream is over. But again, as I said before, the 3 components of a nucleotide are a sugar, a 5-carbon sugar, a phosphate group, and a nitrogenous base. So here's that 5-carbon or pentose sugar in brown, the phosphate group in blue, and then the nitrogenous base in green. These are connected by covalent bonds. It forms a linear molecule with 5' and 3' ends. And then these nitrogenous bases lie perpendicular to the sugar phosphate backbone. So we see our sugar phosphate backbone here, right there, and then our nitrogenous bases are pointing out perpendicularly here. I actually want to go back because I just was thinking of something I wanted to point out on this slide here. I didn't go into a little bit more detail on this diagram. I wanted to. So a nucleotide is, like we said, a 5-carbon sugar, a nitrogenous base, and a phosphate group. Okay, so really, this nucleoside monophosphate, okay, is a nucleotide. Okay, so is this nucleoside diphosphate. So is this nucleoside triphosphate. Depending on the specific base that is in here, this can be adenine triphosphate, right? Or adenosine triphosphate, ATP. It can be guanosine triphosphate, cytosine triphosphate, thymine triphosphate, etc. So depending on this base, that's what's going to determine the name. But when we talk about nucleic acids, I did not want to ignore ATP because ATP is part of this nucleic acid group. Okay, going back to this DNA RNA comparison over here. Okay. All right, so there are some basic structural differences between DNA and RNA that I'm going to highlight today, and then these again will be reiterated in the Unit 6 review, I'm sure. This image from ThoughtCo does a really nice job of illustrating these differences. So the first one is the specific 5-carbon sugar. So, so the deoxyribose sugar is our 5-carbon sugar in DNA. The ribose sugar is our 5-carbon sugar in RNA. The only difference is that the ribose sugar has 1 more oxygen than deoxyribose. So we see here, this deoxyribose has a hydrogen. This ribose in the same position has a hydroxyl group. Another major difference, RNA contains uracil as one of its bases instead of thymine. Okay, and then finally, DNA is usually double-stranded, and RNA is usually single-stranded. So we see that very clearly right here. All right. Okay, so here are some example release FRQs. I'm glad we got here about 10 minutes left. I pulled these actually from AP Classroom, from the publicly available category. So these are FRQs that you can probably, you can, I'm sure, search for on the internet and find the available scoring guides if you wanted to see exactly what College Board has to say about the responses to these particular questions, but here are some examples of the type of questions you might be asked in the context of the FRQs of this year's exam. So, first one, many biological structures are composed of smaller units assembled into more complex structures having functions based on their structural organization. For the following complex structures, describe the smaller units, their assembly into the larger structures, and one major function of these larger organized structures. So any one of these molecules here could be used in a question like this. You can think about how this might work for an enzyme, or for a phospholipid, or for a carbohydrate, or for DNA or RNA. So let's just do the enzyme, for example. So the question asks, describe the smaller units. Okay, our smaller units for an enzyme, we're going to assume that the enzyme is a protein. We're going to say the smaller units of this enzyme are amino acids. We're going to say that amino acids have a nitrogen bonded to two carbons. We're going to say that the amino acid has an amino group on the one end with nitrogen bonded to two hydrogens. We're going to say it has a carboxyl group on the other end. We're going to say the central carbon is bonded to a hydrogen and then to a variable R group that has different structure depending on the specific group. Then discussing assembly into larger structures. Discussing assembly into larger structures, now you talk about dehydration synthesis reactions. You talk about how when you remove a water molecule, you can join two amino acids together via a peptide bond. And then you also talk about the 4 levels of protein structure, how the primary level of structure is the sequential order of amino acids, how the secondary structure is the formation of the alpha helices and the beta pleated sheets from hydrogen bonds formed between atoms in the backbone of the polypeptide. Then that tertiary structure, that the globular shapes are formed from non-covalent interactions between the atoms in the R groups and how the hydrophilic functional groups or R groups will group together in hydrophobic. R groups will group together in hydrophobic interactions. How some charged R groups will form ionic attractions, and so on and so forth. And then one major function of these larger organized structures, like I mentioned, proteins have so many structures. They provide so many functions. They provide structural support. They can be used as signaling molecules. They catalyze reactions, and so on and so forth. Another example FRQ, describe how the properties of water contribute to plasma membrane structure. So here you're going to want to tie together the structure of the plasma membrane, which again, Lee said she's going to go into tomorrow, with these properties of water that we talked about, and the existence of hydrogen bonding between water molecules, and the properties of cohesion and adhesion, and so on and so forth. And then finally, that last FRQ example, the physical structure of a protein often reflects and affects its function. Describe 3 types of chemical bonds or interactions found in proteins. For each type, describe its role in determining protein structure. This is sort of a flip of the answer I described for that first question. So now instead of focusing on this is primary structure, this is secondary, this is tertiary, this is quaternary, now you're just going to focus in on bonds. So here you can talk about covalent bonds and how they form the peptide, or they are the peptide bonds that form that primary structure. You can talk about hydrogen bonds and how they play a role in both secondary and tertiary structure. You can talk about any one of those non-covalent interactions, either ionic attractions, hydrophobic interactions, disulfide bridges. Those are all examples of those non-covalent interactions found in proteins. Okay, so this was the last slide that I had of material that I wanted to share with you. So I'm just going to take a quick look in the chat to see what's going on in the question. Does quaternary structure form off of non-covalent interactions like tertiary? So yes, quaternary structure and tertiary structure both result from non-covalent interactions, yes. If there are any other questions at this point, I would love for you guys to type them into the chat. I'll just wait for like a half a minute or a minute and just see what comes in and try to answer some questions live. Can you go over the first FRQ again? Sure. Okay. So I went through the first FRQ relative to enzymes. Let's do carbohydrates this time. So we're going to talk about the smaller units of carbohydrates. These are monosaccharides. Like I mentioned before, these are typically 5 to 6 carbon structures. Also bonded, these carbons are also bonded to hydrogen and oxygen. When they assemble into larger structures, they undergo dehydration synthesis to form glycosidic bonds, and that makes up disaccharides or trisaccharides or very, very large polysaccharides. Now, the larger organized structures, the function of these structures varies depending on the larger structure itself. If it's a disaccharide or a trisaccharide, it's going to be more of a source of quick energy, right? Those disaccharides and trisaccharides will be broken apart inside the cell to provide the fuel for cellular respiration, for example. A larger, much more complex polysaccharide like starch or glycogen, for example, those are going to be used more for energy storage because it's so many molecules, so many monosaccharide molecules bonded together. And like I mentioned, those linear chains of monosaccharides often get cross-linked together as well. So they're not as easy to, it takes a lot more energy to break those into their individual subunits, into their monomers in order to use them for energy. And so those are good for storage. And then other very complex carbohydrates like cellulose, for example, those are used for structural integrity. Let me take another look at some of these questions. Hydrogen bonds are not covalent. Hydrogen bonds are an example of a non-covalent interaction. Like I mentioned at the very beginning, the word bond is kind of a misnomer in that name. It might actually help you to think of it more as a hydrogen interaction. Let's see. What is ATP's assembly if it does not work like RNA or DNA? I'm going to go back here again. Here. So here's the structure of a nucleotide that will be used in DNA or RNA. It's basically, if this was adenine, which it is here, right? This is adenosine monophosphate. It's the adenine nitrogenous base, it's a 5-carbon sugar, and it's one phosphate group. ATP is adenosine triphosphate. So there's just going to be 2 more phosphate groups attached here. So that's the difference between those 2. Let's see what else. Why wouldn't water break those bonds? Unfortunately, I don't know the context of that question, so I'm not sure which bonds you're referring to. Let's see. Okay, can you explain the second one? Sure. Let's go back to that last one. Okay, describe how the properties of water contribute to plasma membrane structure. Okay, well, I briefly touched on this. Even with the lag, I kind of want to turn it to you guys and see what you might say. So what do you think? How do the properties of water, just very briefly, just share your thoughts, how the properties of water contribute to plasma membrane structure? Let's see what you say in the chat. Okay, it gives shape. The properties of water give the plasma membrane its shape. Yes, absolutely. We talked about how phospholipids are amphipathic, and so they have that water-loving or hydrophilic and that water-hating or hydrophobic region, right? So those hydrophobic tails, those fatty acid tails, they're going to group together. They're going to form hydrophobic interactions to make the inside of the phospholipid bilayer. I'm going to go back to that slide because it's easier if I have something to point to. Here we are, right? So these hydrophobic interactions are all these fatty acids interacting with one another here, and then those hydrophilic or water-loving heads, right, that are made that way by the charged nature of the phosphate group, those orient themselves out toward the water. So the property of water that is driving the structure of the phospholipid bilayer in this example is the fact that it's polar, that it's a polar molecule. Yes. Okay. So a lot of people saying similar things. Clifford's advice is great. Attack this question from either the water side of the question or the membrane side of the question. Yeah, these are all excellent ideas. Yeah. So you're really getting the hang of it. That's awesome. All right, so I don't want to keep you longer than the hour. We said these were going to go an hour, so I'm going to end this here. But thank you so much for coming. I really hope this was helpful for you. Stay safe and stay healthy. Good luck on the AP exam. And if you have any questions, please tweet at me. I'll be happy to answer them. The next session is tomorrow at 2 p.m. It's going to be on Unit 2, Cell Structure and Function. The information is linked below the video. Thank you again. Have a fantastic rest of your day.